In this video, we will discuss the advantages of using pSIVA to study the kinetics of phosphatidylserine externalization during apoptosis. The distribution of phospholipids in normal eukaryotic cells is asymmetrical, and phosphatidylserine primarily resides in the inner leaflet of the plasma membrane. During the early stages of apoptosis, phosphatidylserine flips from the inner to the outer bilayer. Phosphatidylserine externalization is considered a hallmark of apoptosis, involved in a variety of physiological and pathological conditions. Classic reagents to detect phosphatidylserine translocation, such as Annexin 5-based probes, have limitations. One limitation of using Annexin 5 fluorescent conjugates is that they fluoresce when free or bound to phosphatidylserine and require a wash step to reduce fluorescence background. When using the advanced Annexin 12-based probe pSIVA, there are no wash steps. This is because pSIVA, conjugated to IANBD, is a polarity-sensitive dye capable of on-off fluorescence. In solutions such as media, the fluorescence of pSIVA is off. However, when pSIVA is bound to exposed phosphatidylserine in the membrane, a nonpolar environment, it fluoresces. This novel property makes pSIVA ideal for tracking phosphatidylserine externalization in real time. For instance, phosphatidylserine translocation is clearly visualized by an increase in pSIVA staining in a model of neuronal cell death. Real-time monitoring of transient phosphatidylserine exposure is also unique to pSIVA. If phosphatidylserine flips back to the inner bilayer, pSIVA is released and results in the loss of fluorescence. In the second clip depicting recovery from neuronal degeneration, an initial increase in pSIVA staining is followed by a reduction in fluorescence for cultures in which nerve growth factor was added back. A decrease in fluorescence indicates less pSIVA is bound to phosphatidylserine and suggests these cells have been rescued from cell death. A quantitative evaluation of normalized pSIVA fluorescence in rescued versus nerve growth factor deprived neuronal cultures facilitates the identification of individual neurons rescued from cell death and helps to define the critical window for recovery. pSIVA has been used to analyze phosphatidylserine exposure in various cell cultures and primary cells in flow cytometry and microscopy, as well as for in vivo and tissue applications. Due to its low background staining, pSIVA demonstrates sensitive detection of permanent and transient phosphatidylserine translocation. Learn more about the benefits of pSIVA and how it can advance your research at novusbio.com.